today's lecture we will summarize the course, and in doing so we will consider all the functions and the characteristics of the biomolecules that we have studied during the duration of the course. Now initially when we started off, I showed you the different topics that we would be covering, namely structure and functions of biological molecules. All these molecules, biological macromolecules that play an important part in living systems and how they function. We and proteins went on to doing enzymes, followed by vitamins, coenzymes, carbohydrates and lipids, and then looking at nucleic acids and their components, that is DNA and RNA, and what they were comprised of and their specific characteristics, which led us on to do the principles of bioenergetics and basically carbohydrate metabolism, where we considered the breakdown of glucose. In this summary lecture, I will be going briefly over all the topics that have been covered here. Now, when we consider the biological functions, the basic need is to understand the structure, which means that obviously an understanding of a biological macromolecule, be it a protein, an enzyme which is a part of a protein, which is required for all the specific processes, reactions that go on in the body, you understand that we have to have a knowledge of its structure, because only with a complete understanding of its structure do we know what specific functions it can accomplish. If we look at the basic amino acid structure, which is something that you all know by now, we have attached to it an amino group, a carboxylic group and a side chain R group, which actually is what gives it the specific function, specific characteristics that make it or allow it to perform certain reactions and undergo certain changes that are going to bring about the reactions that go on in our body, the chemical processes that go on in our body. The linking of the amino acids together gives rise to a peptide bond that is the basics of any protein structure. So here we have a dipeptide that is linked by two R groups, and we know that these R groups can have a varied number of properties depending on the components present in the R group. They could be negatively charged, they could be positively charged, they could be uncharged and yet polar, and they could also be hydrophobic in nature, non-polar in nature. And we know that on folding of a protein, which we will just overview in a minute, we know that these non-polar components, the non-polar amino acids, would likely be at the center of the protein, in the core, the hydrophobic core of the protein. In considering the different residues, the amino acid residues, we also learnt of their 3-letter codes and their 1-letter codes that were used to designate the amino acids linked one after the other, since we know that each of them are going to be linked by a peptide bond. Now, in understanding how these residues actually behaved in globular proteins as opposed to membrane proteins, we considered that all these non-polar residues, the ones that are hydrophobic in nature, would in a globular protein rather remain in the hydrophobic core of the structure than on the surface, which is the case for a membrane protein. Because when we went on to study the lipid bilayer, we knew that when this integral membrane protein had to traverse the lipid bilayer, it so happened that the core would have to be hydrophilic in nature so that it could transfer the ions, but the surface amino acid residues would have to be hydrophobic in nature so that they could interact with the lipid fatty acid chains. However, for a globular protein which is interacting with a polar solvent, in a polar environment, it is most likely that the polar charged residues would be on the surface of the protein, and these would comprise most of the catalytic sites that would bring about the reagents or the substrates to the enzymes for a particular reaction. And the non-polar hydrophobic amino acids would prefer to reside in the core of the protein. In looking at the protein architecture, we considered the sequence of the protein, which was thus the linking of the amino acids one after the other to form specific secondary structures that are folded amino acid sequences having definite hydrogen bond characteristics, definite areas in what we call the Ramachandran plot. After the formation or the understanding of the secondary structure, we consider the tertiary structure of a protein, which is the native folded form of the protein, and in some cases there may be an association of subunits leading to a quaternary structure of the protein, for example like in hemoglobin. In the conformational flexibility that can occur in proteins due to the rotation about the single bonds, the peptide bond we know is partially double bond in character because of the lone pair of electrons on the nitrogen atom. We have a peptide plane that renders this less prone to any flexibility, even though we do see cis peptides and mostly we observe trans peptides, so that we do not have the R groups that may be bulky in nature actually clash with one another. But the rotations are possible about the NC and the CC bonds that give rise to what are called the phi psi angles, and we learned from the Ramachandran plot there are definite designated areas for the right-handed alpha helix, the left-handed alpha helix and the beta sheet of the protein, which identifies specific secondary structural elements of the protein on its folding. Considering other aspects of protein folding, this is one very particular example that we consider that is now known as the famous Anfinsen's experiment, where the protein sequence, the importance of the protein sequence in understanding the protein structure was emphasized. In this case, the ribonuclease A molecule, which happens to have 8 sulfide containing amino acids, that is 8 cysteines that have the possibility of forming 4 disulfide bonds, was actually reduced in a fashion where we see that these linkages bring different parts of the amino acid sequence together by virtue of the formation of a disulfide bond. So, when we have a cysteine formation from 2 cysteines joined by disulfide bonds, we can then break them up with the specific reducing agents. Now, if that happens, what we can actually, in Anfinsen's experiment, what was done, open up this chain to result in 8 such SH, 8 such cysteines, and then allow the protein to fold by removing the reducing agent as well as the denaturing agent, and on removal it was found that the exact same bonds or the same disulfide bonds were formed, which was actually considered or determined when the same activity was retained for the protein, which meant that it had its final folded native structure. So, in this experiment, after we have the native structure correctly formed from the 4 disulfide bonds, we know that all the information that is required for the protein to fold is actually present in its amino acid sequence. But this is the protein folding problem, where we still cannot predict how a specific sequence goes to this single conformation in its native structure. There are certain methodologies that actually go into understanding the folded protein structure, and we know, apart from specific guidelines or specific presence of secondary structure where we have hydrogen bonding present, there are certain energy terms that can account or do account for the total energy of a protein, and we know that it would rather go to a minimized energy conformation. We have covalent interactions and non-covalent interactions, and the minimization of all these interactions together to give an optimum energy would give us the actual energy or the actual condition that the protein would likely to be in. So, we have a stretching energy, a bending energy, a torsional energy that is concerned with linked atoms. We have non-covalent interactions where we have the van der Waals interaction, Coulomb interaction and the hydrogen bond interaction. Together, from the different energetics that are involved in the formation of these specific interactions, be they covalent or non-covalent, we have a specific force field that gives us the total energy of the protein, and a minimization of this considering certain variations in the bond length or in the angle or in the dihedrals and certain energetic references with regard to non-covalent interactions would give us the total energy of the molecule, which actually leads to the energy minimized structure, which would be wanted to predict from the primary sequence of the protein. So, the features of the native state say that there is a well-defined 3D structure and there is a characterized molecular function with this, and the idea would be to get from an unfolded structure a total amino acid sequence to the folded structure. So, many proteins actually fold spontaneously to their native structure, which results in a very fast process, but the predictive methods are still not very accurate into determining what the final structure might be. And there are also other proteins called chaperones which actually help certain proteins to fold to their native form. They assist the proteins in folding into their native form. So, we considered in the first part of the course where we had our individual amino acids, we considered the various properties of the amino acids and then how they linked up to form the overall protein structure. Now, once we have the protein structure or we understand the characteristics of the different side chain residues and what reactions or what types they actually were, then we know what other substrates may interact with them into giving their chemical reactions or their processes or the enzymatic reactions that are extremely important in all biological processes. So, if we consider these thermodynamics of protein folding, there are certain aspects of the protein folding that give us what is called a free energy funnel that have a total number of conformations, possible conformations leading to a single native structure. We then went on to understanding enzymes. Different types of enzymes. We considered that there were 6 classes of these enzymes that actually had specific reactions that they catalyzed, because we know that these enzyme catalysts actually reduced the free energy of folding or the free energy of the reaction, where we would have a favored reaction that would have less activation energy due to the presence of the catalyst, which is the enzyme. So, the 6 different enzymes possible are oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. Each of these have their own characteristics. There are specific amino acids that are going to be important in bringing about these reactions. For example, there are many reactions that we consider called transferases, and a specific example of these are kinases that actually transfer phosphate from ATP to other molecules, which is an extremely important process in the metabolism or metabolism pathways. Now, if we consider the amino acid residues that actually can or do form different forms in the sense that they can act as an acid or as a base, and in that case they may be a proton donor or a proton acceptor. Now, this becomes extremely important when we consider different enzymatic mechanisms, as we had seen when we studied the mechanisms of ribonuclease, lysozyme and chymotrypsin, which is a protease. Each of these enzymes had specific amino acids at the catalytic sites and because of their presence and because of their capability of acting as proton donors and proton acceptors, they could bring about certain reactions in the substrate, transforming them to the product. In the process, the enzyme which acts as a catalyst does get back to its original form, so that it may accept another substrate to form into the product again. In this process, we considered Michaelis-Menten kinetics, where we considered the velocity of the reaction with substrate concentration, and we know that at low substrate concentration we have a first order reaction with respect to the substrate, and as the substrate concentration is increased, we have the reaction independent of the substrate concentration becoming zero order with respect to S, because the enzyme has a limited number of active sites which can accept the substrate molecule. Now, from this we studied the maximal velocity, the Vmax, which is the maximum velocity, the maximal rate of the reaction, which is actually never achieved in reality, but is nevertheless useful for specific understanding of the reaction. And in this case, to reach the Vmax, we understand that all the enzyme molecules have to be involved or tightly bound with the substrate. Then we consider Michaelis-Menten conditions, where we derived a Michaelis-Menten constant, a Km value meaning tight binding during the turnover, and a high Km meaning weak binding during the turnover, and we looked at specific expressions which we got from Michaelis-Menten kinetics that actually related the Km, the substrate concentration, and the maximal velocity of the reaction, the Vmax. Now, considering the Vmax and the Km and the Kcat values, we consider the 4 different types of enzyme inhibition that are possible. We have irreversible inhibition that renders the enzyme inactive for any further reaction. We have competitive, non-competitive, and uncompetitive, and in each of these cases the enzyme or the enzyme substrate complex has some interaction with the inhibitor, and we can look at a specific inhibition constant considering the enzyme and inhibition concentrations. We looked at then Lineweaver-Burke plots that plotted 1 by V versus 1 by S, giving us straight lines for the uninhibited enzyme, and for specific types of inhibitors we observed specific types of graphs. The y-intercept in this case gave us 1 by Vmax, and on the x-axis we got the value of minus 1 by Km for each of the cases, which we could consider based on the Lineweaver-Burke plots for the different types of inhibitors. We got a case where for the competitive inhibitor the same Vmax was achieved. So, we knew that this characteristic where we had the same y-intercept would mean that this was a competitive inhibitor. We had a non-competitive inhibitor, the Vmax changed but with the same Km, and we had an uncompetitive inhibitor when we had parallel lines. So, this actually summarized the enzyme kinetics portion, from which we consider specific active sites of enzymes, the specificity of the enzymes which we studied with relation to ribonuclease, lysozyme, and chymotrypsin. We then went on to do phospholipids, considering glycerol. Glycerol, when linked with fatty acids, forms the triacyl glycerols, which were the storage lipids. The membrane lipids that are polar in nature have, apart from 2 fatty acids linked to them, a phosphate and an alcohol linked to it, which would be the glycerophospholipids. We had the sphingolipids, and we had glycolipids, where we had a sugar group attached to the lipid as well. Then we looked at the different molecular arrangements of the lipids that could actually form micelles, liposomes, or the bilayer sheet. Now, in consideration of the bilayer sheet, since this formed the cell membrane, we also had transferred across the membrane. So, we studied the specific proteins that were embedded in the membrane, where we would have peripheral proteins, integral proteins, or ones with a lipid anchor. We then went on to consider the fluid mosaic model. The fluid mosaic model actually gave us the idea of how the transportation of material went from the cytosol to the inner part of, say, the mitochondria, or from the outer part of the cell to the inside of the cell. And we learned there were specific ion channels that would actually facilitate the diffusion of ions or other components in and out of the cell. And each of the traversing helices we consider were important in their structure, in their characteristics. We also found that there were beta porins, porin molecules that actually formed a beta barrel structure that would also account for the transfer of material inside and outside, from the extracellular to the intracellular, or from the intracellular to the extracellular. And we looked at specific proteins that were involved in those cases. In considering membrane transport, we also looked at the specific electrochemical potential that is involved in the process, where we have a concentration gradient along with a membrane that has a membrane potential that is associated with the electrochemical potential of transfer. We then went on to study carbohydrates. Now, in carbohydrates, we considered their importance with respect to their serving as energy stores, fuels and metabolic intermediates, that they form part of the structural framework of RNA and DNA, these sugars, and the structural elements in the cell walls of bacteria and plants, and in the exoskeleton of arthropods like chitin, and that they are linked to many proteins and lipids, where they form linkages such as these are called glycoproteins and glycolipids, and they also play a very key role in cell-cell recognition processes. In understanding carbohydrates, we consider the monosaccharides that were the simplest of carbohydrates, and they being aldehydes or ketones that have 2 or more hydroxyl groups. In this case, we consider the open chain forms of glucose and fructose that actually cyclize into rings, where the aldehyde can react with the alcohol to form an intramolecular hemiacetal, and a ketone reacts with an alcohol to form an intramolecular hemiketal. Now, in these cases, there are certain other reactions that take place, and we also have different conformations possible, where we can have a chair or boat configuration and a furanose ring that adopts an envelope form. In this case, we have the hemiacetal and the alcohol, and we have different sort of linkages that link these two monosaccharide units to form a specific disaccharide, where we have an alpha or a beta linkage. We went on to study these linkages of the alpha glycosidic and the beta glycosidic, and we learnt from the orientation of the OH how such a linkage would be possible, and based on these linkages, how we could have the formation of different disaccharides, and which led to different polysaccharides, where we could have straight chain polysaccharides, we could have branch chain polysaccharides, and depending on the type of polysaccharides that were formed, they would have specific functions. We could have cellulose, that would be a fiber type polysaccharide, or we could have granular like starch, where we would have a granular polysaccharide, and each of these were dependent on the type of linkages that linked the monosaccharide units together. So, we then went on to study vitamins. As we went on to vitamins, we gradually get an idea of how complicated these molecules can actually get in the biological systems, and we looked at a specific series of water soluble vitamins and the lipid soluble vitamins as well. Now, each of these vitamins have a specific role to play in the formation of coenzymes. Now, in the formation of the specific coenzyme, each of these have a specific role in the final reactions that they would go to. For example, if we consider the coenzyme ATP, ATP is extremely important in the specific reactions that it goes by because of the fact that it has a very high energy phosphate bond. In considering this high energy phosphate bond, we know that ATP going to ADP plus Pi can give us an energy of minus 30.5 kilocals per mole. Now, that is extremely important in the driving of certain other reactions that take place in the metabolic processes, as we had studied when we considered the glycolysis, which was the breakdown of glucose. If we consider some of the other vitamins, for example, niacin. Niacin has a role in the formation of NAD and NADP. Now, in this formation, we have it play an important role in redox reactions. Now, why would niacin or why would NAD or NADP actually play a role in a redox reaction? Or why would FAD and FMN play an important role in redox reactions? The reason being that these can act as proton and electron acceptors and donors. So, we have them have an oxidized form and a reduced form. In this reduced form, when they accept the hydrogens, there are certain reactions that require a redox change, and in these certain cofactors or these certain coenzymes that are going to be involved in these enzymatic reactions, which result in a redox process going on in the body. So, we have redox reactions that are usually accomplished by NAD and FAD or FMN. So, considering the type of reaction that is involved, each of these vitamins that form the coenzymes have a specific role to play in the biological processes that actually go on. For example, when we consider pantothenic acid, that is crucial in the formation of coenzyme A, which is extremely important in the acetyl coenzyme A component that is required for the glycolytic pathway that gets into the Krebs cycle later on. So, all of these cases where we consider each of these vitamins, the roles are extremely important, and the roles being possible, the vitamins being transformed into the coenzymes, and each of these coenzymes having a specific role to play in a biological process. For example, when we consider thiamine, which forms thiamine pyrophosphate TPP, which results in the transfer of 2 carbon atoms. When we consider pyridoxine, we have PLP, we have lipoamide and ubiquinone, again another electron carrier, lipoamide as an acyl transfer. So, each of these roles, as we consider the specific examples in bioenergetics and in metabolism later on, we understood how each of these were actually extremely important for the specific function that they were assigned to. In considering nucleic acids and nucleic acid structure, then we considered the sugar phosphate backbone. Now, this is the primary structure or the primary idea of how the nucleic acids are formed. The backbone is comprised of sugar and phosphate, and we have specific bases attached to the sugars, and this sugar and phosphate backbone is different in DNA and RNA, the sugars being different and the type of base attached being different. Now, the two major types of nucleic acids, DNA and RNA, we had specific sugars that were used. Considering the specific sugars that were used, we had ribose that was used in RNA, deoxyribose used in DNA. Now, the difference in this was the specific OH at the 2' position that was missing from the DNA sugar molecule. So, when we consider these linkages, we now know that in case of the absence of the OH group from the 2' position, we know that this forms a deoxyribose that is used in DNA and the ribose that is used in RNA. The linkages are through the phosphate at this position, and we have a base attached to this position. The nucleic acids are made of polymers. They are extremely important in the specific structure and how actually proteins are formed from nucleic acid structure. We have the central dogma of biology, which is DNA to RNA to protein. So, an understanding of the nucleic acid structure and what comprises their structure is extremely important in understanding how the specific reactions take place, or the specific, we will look at the bases in a minute right now, where the bases actually define which amino acids are going to come one after the other to comprise or to form this primary sequence of the protein, of the polypeptide chain, and it is this protein that is going to fold into a specific structure, a specific enzymatic structure, and bring about a certain reaction. So, an understanding of the basic level, the base level, is important. In RNA, we now know that the RNA and the DNA have different sugars that are formed. We have the use of AMP, CMP, GMP and UMP. In DNA, we use the deoxy forms of AMP, CMP, GMP and TMP, where the small d refers to the fact that this is a deoxy form, and the 2' functional group is what is different, as well as the uridine or thymine as the 4th base. Thymine is what we find in DNA and uridine is what we find in RNA. So, what are these guanine, cytosine, uridine and thymine? They are what we call the bases, the nitrogenous bases that are comprised of 2 families, the base families, that are the purines and the pyrimidines. In considering the purines, we have fused 6 and 5 membered hetero CN carbon nitrogen systems that are usually unsaturated, and the 2 common purines in biological systems that are used in DNA and RNA are adenine and guanine, which are the purine, forming the purine family. If we consider the pyrimidine family, we have a 6-membered carbon nitrogen ring that is also unsaturated mostly, and the 3 common purines that we see here are cytosine and thymine that is used in DNA, and cytosine and uracil that is used in RNA. So, if we consider the purines and the pyrimidines together, we have adenine, guanine, cytosine, thymine and uracil. The 6-membered carbon nitrogen ring group being formed from the pyrimidine family, and we have the adenine and guanine formed from the purine family. And it is the linkage of these to the sugar ring, at this position, that is going to result in different formations and different linkages. These different linkages or different order of bases is going to result in a different form of a protein, because a different amino acid codes for every triplet codon, as I will just mention again in a moment. So, what we have in DNA is we have the sugar phosphate backbone, and all of us remember now that this sugar is lacking the OH group at this position. So, we have the sugar phosphate linkage that forms the backbone, and we have each of these sugar rings linked to a specific base. The bases belong to either the purine family or the pyrimidine family. The important thing here is the understanding of how the bases link to one another if we are to understand the double-stranded, double helical structure of DNA. When we have the linkages, we have a purine linked with a pyrimidine in each case. So, we can have what is called an AT linkage, or we can have a GC linkage. The interesting part of this is that in the formation of a linkage between a pyrimidine and a purine each time, we have a constant distance that we learnt in the classes was around 11 angstroms from one point to the other, from the C1 carbon of say the purine in this case to the C1 of the pyrimidine in this case, and that is also 11 angstroms for this particular linkage. So, it becomes extremely important in this ladder representation or in the formation of the double helical structure of DNA. In the formation of the helical structure, we have certain interactions that are very important here. So, apart from the fact that we have the pyrimidines and the purines linked with one another, where we have one purine linked with a pyrimidine, G linked with C and A linked with T and so on and so forth, we also have specific number of hydrogen bonds in each case. For example, when we have an AT linkage, we have 2 hydrogen bonds, whereas when we have a GC linkage, we have 3 hydrogen bonds. Now, what this does is this imparts a certain stability to the DNA depending on the number of hydrogen bonds that we are going to have, be they 2 or 3 in nature, which then leads to specific stability, which I will show you in a moment again. In the RNA structural elements, we usually have a single stranded or sometime double helical or an unpaired loop or a bulge, or because we do not have a specific double helical structure like DNA. But nevertheless, there are certain linkages that might also be possible between the purines and the pyrimidines in the formation of the hydrogen bonds. So, if we compare the structures, we have the sugar of DNA as a deoxyribose, a ribose sugar for RNA, and we looked at the specific bases that are possible, the strands and the formation of a helix. If we now look at a specific type, the formation or the presence of a specific number of hydrogen bonds, we can look at what is called the melting temperature of DNA that tells you exactly the percentage of the GC content that you might have, which we have certain factors that actually are important in determining what the melting temperature of the DNA is. Now, in considering these different structures, DNA to RNA to protein, there is something else that we also mentioned in class, where we consider that when we go from DNA to RNA to protein, each of these RNAs, they have the messenger RNA that brings a specific transfer RNA that has the anticodon for a specific amino acid that is going to form a polypeptide chain. This in the ribosome assembly actually links up one amino acid with another through a peptide bond, forming the polypeptide chain. And this folded protein, the polypeptide chain which forms a folded protein, is how proteins are synthesized in the body from the biosynthesis of proteins that is going to eventually give us the enzymes that are responsible for all the reactions. We then went on to the last chapter of the course that involved biochemical energetics. Now, in the process of understanding all the energetics or all the metabolism of the carbohydrates, we consider very important reactions that actually go on in the body. We have the oxidative phosphorylation, which is the process in which ATP is formed. Now, ATP is the energy currency of the body, and it is extremely important that the understanding of this oxidative phosphorylation, which is an aerobic oxygen requiring process, will actually occur by a series of electron carriers that ultimately transform the electrons to resulting in the reduction of O2 to water. Now, in all of the reactions in energetic processes that go on in the body, no energy goes to waste. For example, if there is a particular reaction that is not required to occur all the time, in that case, nature has chosen such a very beautiful way of adjusting this property. When we have the free energy of this particular reaction, it is positive, making it non-spontaneous in nature. But what happens is, this reaction is now coupled with another reaction that happens to have a high negative value, a negative delta energy that more than compensates for the positive value required for the specific transformation. Now, as soon as this transformation is required in the body, the ATP or whichever high energy bond is going to come to provide the energy will come into the picture, and these two reactions will be coupled together, and in the coupling the non-spontaneous form will then be able to form its product that will then go on to the specific steps of the glycolysis cycle, as we will look into the specific example of glucose forming glucose 6-phosphate. So, what happens is, since the free energy changes of the coupled reactions are additive, we have two coupled half reactions where one of them is spontaneous and the other is non-spontaneous. Now, at the enzyme active site, what happens is the coupled reactions are kinetically facilitated while the individual half reactions are prevented from occurring. For example, if a certain reaction would have a negative free energy, then it would so happen that this would, since it is spontaneous, it will continually occur. But this also does not occur because it is an enzymatic reaction. It has to go to the specific enzymatic site for this release to occur. But what happens is the enzymes do not look at these half reactions. They would rather have the coupled reaction which is kinetically facilitated, but the half reactions which are not. So, the free energy changes of the half reactions are summed to yield the free energy of the coupled reaction in various complicated processes that actually go on in the body. We looked at the specific diagram here of the mitochondria, where we know that we have proton pumps, we have specific proton concentrations on the inside and the outside of in the intermembrane space and in the inner central region of the protein, of the mitochondria rather. So, if we look at the intermembrane region and we look at the outer membrane and the inner membrane, we know that there are specific complexes involved with certain reactions. Now, each of these complexes actually are comprised of a set of enzymes that bring about certain reactions, which ultimately leads to the formation of ATP that is driven by a proton motive force. So, what we have is we have a specific chemical potential that arises due to the difference in the hydrogen and ion concentrations in these two regions. We also have an electrical potential that is due to the difference in the membrane potential here, which results in a proton motive force that ultimately leads to ATP synthesis. We have therefore a delta G associated with a change in pH, because there is a difference in the hydrogen ion concentration. We also have a change associated with the membrane potential. We have the intermembrane space and we have the membrane space, and we have a proton pump that actually pumps the protons from this inner space to the intermembrane space, which actually already is high in concentration of protons, but nevertheless we need this high concentration of protons to drive the ATPase that is going to result with the protons in the proton motive force to form ATP, and ATP we know is required in a lot of the reactions that go on in the glycolytic cycle. So, what happens here is we have a specific rotation of this stalk that results in a specific conformational changes of the beta subunits here, and these beta subunits, the catalytic sites, will result in the formation of ATP from ADP and PI. So, essentially what happens is we have the formation of ADP plus PI in a process with electron transport and oxidative phosphorylation to form ATP. In this, what happens is we have FAD and NAD plus, which actually is formed from the vitamins. We know these are formed from the vitamins niacin and riboflavin now, and we have protons and electrons come into the picture, where we have the reduced form that then with the electron transport forms a oxidized reduced cycle, and this is accomplished by a series of enzymes. And the overall reactions in oxidative phosphorylation that ultimately lead to the reduction of oxygen to form water. In the process, it forms ATP. In the formation of ATP, for every NADH that we get approximately 2.5 moles of ATP, and from FADH2 we get 1.5. In the catabolic processes that we considered in our metabolism, we considered the breakdown of glucose in this case. But if we look at the overall picture where our consumption, our food consumption involves lipids, polysaccharides and proteins, each of these break down into smaller units, fatty acids, glycerol. We have fatty acid oxidation, glycerol getting into here. Then we have polysaccharides that break down into monosaccharides, finally get into glycolysis cycle. We have proteins that break down into amino acids, and with further amino acid breakdown, we finally get into the pyruvate. That is the final step of the first cycle, the glycolysis cycle. The pyruvate then forms acetyl-CoA. We learned coenzyme A formed from pantothenic acid, which is a vitamin. Then from the acetyl-CoA, it goes into the TCA cycle. Offshoot of the TCA cycle being the formation of oxidative phosphorylation with the utilization of NAD and FAD, where we have the formation of ATP, and in the glycolysis cycle or the glycolytic cycle also there are steps that involve the production of ATP. The final breakdown of the glucose is going to lead us into the formation of carbon dioxide and water. In the glycolysis cycle, these are the certain steps that actually go on into this, where we are considering the breakdown of glucose, and we learnt in the process of understanding these glycolytic steps that, for example, in the first step where we have glucose going to glucose 6-phosphate, the delta G associated with this reaction is positive. But the breakdown of ATP to ADP plus PI results in a large negative delta G. So, these reactions are coupled together in the enzymatic site of hexokinase. Hexokinase will then act on these two coupled reactions together to give glucose to glucose 6-phosphate. And these specific reactions that involve kinase, we know, involves the transfer of the phosphate moiety. For example, the hexokinase or the phosphofructokinase and so on. What happens in these cases is these specific steps are irreversible. As a result, when glucose is trapped into the cell into forming glucose 6-phosphate, it usually does not get back into forming glucose. So, once the glucose is trapped in this glycolytic cycle, it will go on into considering the breakdown of glucose, which is finally going to result in our energy, formation of carbon dioxide after the TCA cycle. So, we have our glucose go to glucose 6-phosphate, this go to fructose 6-phosphate, fructose 1,6-phosphate, where we actually have the breakdown from the 6 carbon unit to the 3 carbon units. Then when we continue this, we have, there are 2 glyceraldehyde 3-phosphates per molecule. So, this actually occurs twice, and this is the step where we have ATP formation, and this is another step where we have ATP formation, and we have the formation of pyruvate starting from glucose. It is this pyruvate that then goes on into the next step. So, when we have glycolysis, we have this particular step where we have glucose getting into pyruvate. The formation in fermentation, we have glucose forming lactate, where the anaerobic catabolism will give us actually 2 high energy bonds of ATP. So, if we look at the glycolysis enzyme reactions, we have hexokinase, phosphofructokinase and pyruvate kinase. Each of these has a specific delta G0 prime associated with it, a delta G0 prime meaning that we are considering the cellular conditions, cellular or rather body temperatures into calculating the delta G0 values, where the temperature we consider is not 25 degrees centigrade, but 37 degrees centigrade. So, we have our hexokinase, phosphofructokinase and pyruvate kinase that result in large negative delta G values. So, and it is these reactions that are actually irreversible in the steps. Following this, we go for glycolysis. Glycolysis is followed by the Krebs cycle. So, this is actually what occurs in the cytosol, then this is what occurs in the mitochondria. We have the glycolysis step, the glucose forming 2-pyruvic acid, we have this pyruvate going into acetyl-CoA then leading into the Krebs cycle, And after the Krebs cycle, the reactions that we get from there, NAD, NAD plus, mainly by NADH, the electron transport that finally leads into the formation of ATP. So, we are looking at energy production. So, the breakdown of glucose is going to give us energy. So, we have the Krebs cycle where the acetyl CoA comes into the picture. We have the formation of NADH, a formation of ATP, formation of FADH2 and NADH, which gets on into the electron transfer processes of oxidative phosphorylation, and we have the formation of carbon dioxide that results from the breakdown of glucose. So, if we look at the balance sheet for the phosphate bonds of ATP, we have 2 ATP expended, we have 4 ATP produced. So, the total net production of 2 phosphate bonds of ATP per glucose. And in aerobic mechanisms, we have the pyruvate produced in glycolysis, that is oxidized to carbon dioxide via the Krebs cycle. The NADH that is produced in the glycolysis and Krebs cycle, which we saw in the previous slide, where we have the formation of the NADH in these steps and the FADH2, that is, or rather has to be reoxidized, because it is now reduced, it has to be reoxidized. This is reoxidized via the respiratory chain, where additional ATP is formed. So, if we look at the total number of ATP that we actually get ultimately formed from the breakdown of one glucose, these are the number of ATP formed, that are 30 to 32 depending on the specific reactions that we are considering. And for each of these, for these 2 cases where we are forming glucose 6 phosphate from glucose or fructose 1,6 phosphate from fructose 6 phosphate, we require ATP to form the phosphates in a reaction mechanism that involves the enzyme kinase. And then we go on to see the number of ATP or reduced coenzymes that are directly formed in each of these steps, and we get to a final 30 to 32 ATP ultimately formed from one glucose. So, if we look at the efficiency of biochemical engines in living systems, the oxidation of one glucose actually yields this amount of energy, 2840 kilojoules per mole. So, the energy obtained by the biological engine gives us 32 ATP, that is 30.5 kilojoules per mole, giving us 976 kilojoules per mole. And if we consider standard conditions, the efficiency that we are going to get is 34%, but the cellular conditions are such that it gives us an efficiency close to 65%. So, this is considering the energetics of our system with the breakdown of glucose. So, this has been this course Biochemistry 1, where we have considered starting from amino acids, structures of biological macromolecules, to enzymes, to lipids, carbohydrates, vitamins, coenzymes, then nucleic acids, their components, and biochemical energetics. This has given you a brief understanding of how biochemical processes or biological macromolecules, what are present in the body and what their importance is, the role that they play in the functions that go on into the body. And you have to understand that understanding the structure of each of these components is extremely necessary to get a complete understanding of the function that they can characterize, or the functions that they can accomplish, or an understanding of the reactions that they can actually accomplish. Thank you.